Have scientists actually created life? This video explores the boundaries of cellular synthesis, DNA engineering, and modern laboratory analysis techniques used to study the building blocks of life. What does it truly mean to create life? This ranges from synthesizing organisms from scratch and cloning, to genetic modification, or even attempting to reanimate dead cells. Each method carries different scientific, ethical, and technical challenges. To understand life, we must define it. Fundamental characteristics include metabolism, the ability to reproduce, evolutionary adaptation, and the capacity to react to environmental stimuli. The theory of abiogenesis proposes how life might have emerged on early Earth through chemical evolution, leading to the first replicators and eventually complex biological systems. A landmark in this field is the Miller-Urey experiment. It demonstrated how, under conditions simulating the primitive Earth, simple chemical precursors could form amino acids, the building blocks of proteins. The RNA world hypothesis suggests that RNA may have been the precursor to life, serving both as a carrier of genetic information and as a catalyst for chemical reactions. In the lab, scientists are making progress with artificial protocells. Current achievements include the creation of lipid membranes, microscopic vesicles, and rudimentary self-replication systems. In 2010, Craiga Ventera and his team achieved a significant milestone by synthesizing the genome of Mycoplasma mycoides and successfully transplanting it into a recipient cell. Was this truly an artificial organism? There is an ongoing debate. While the genome was synthetic, the experiment relied on an existing, natural cell as the host. Today, our capabilities in DNA synthesis continue to grow, moving from basic viruses and bacteria toward the potential for more complex, fully synthetic designs. Synthetic biology has broad applications. In medicine and pharmacology, it is used for vaccine and insulin production. In energy, it aids in developing biofuels and new metabolic pathways. Can we create a cell from scratch? The current answer is no. While we can integrate synthetic components into living cells, achieving de novo synthesis of an entire cell remains a significant challenge. Major questions still persist in science. Specifically, how the first genetic information, metabolic processes, and self-replication mechanisms originally emerged. Despite these challenges, we have achieved major milestones, successfully designing a full bacterial genome, transplanting it into a cell, and confirming it can replicate. In summary, while scientists have gained the ability to design and modify life, we are still far from fully understanding or replicating the origin of life itself. Looking ahead, the future of synthetic biology raises profound questions about the limits of scientific intervention and our responsibility when engineering new life forms. Ethical considerations are paramount. We must carefully evaluate the biosafety, biosecurity, and the long-term environmental impacts of introducing synthetic organisms into the natural world. As we advance, international regulations and responsible research guidelines will become increasingly critical to ensure that progress remains safe and beneficial for society. This video has highlighted the bridge between ancient chemical evolution and modern synthetic techniques, showing how far we have come in understanding life. We have explored the key milestones, from the Miller-Urey experiment to the synthesis of microbial genomes, reflecting on the complexity of the life creation puzzle. Science continues to evolve. As our tools improve, so does our ability to manipulate the fundamental code of existence, opening new doors for discovery. If you wish to dive deeper into this fascinating field, we have curated a collection of scientific literature, academic publications, and sources for your reference. Our bibliography features key works, including books by Nick Lane, Paul Davies, and Craiga Ventera, as well as foundational research papers and reputable online science platforms. We encourage you to explore these resources to further understand the ongoing research into abiogenesis, synthetic biology, and the profound questions surrounding the origin of life. Thank you for watching. We hope this video has provided a clear and informative overview of the current state of science regarding the creation of life.